All right, ladies and gentlemen, microbiology and disease. So, what type of things cause infection? Name me one. What? Dirt? What's in the dirt, though? Dirt's inanimate. Bacteria, so that's one thing. What else causes infection? Fungus, very good. What else? Who? Parasites, okay. Not parakeets, parasites. What else? Bacteria, fungus, parasites. What's the flu? Virus. Okay, so you are a host, you are a biome, you are an ecosystem for more non-human cells than human cells. There's more bacteria, fungus, virus, whatever on you, in you, around you than you, right? So you are an ecosystem. So the stuff that's in you, on you, and normally is your normal flora. So flora is like the vegetation that grows. So this is the bacteria and the stuff that grows on you. And as long as it's there in its place, it's beneficial. Like all the bacteria in your gut. You have like 5 to 10 pounds of E. coli in your gut. And what they do is they help you break down food and they release nutrients, vitamins, and help you absorb them as well. Now, if you kill off your normal flora, one of their job is to keep the bad stuff at bay. So there's certain drugs that if you take, it'll disrupt your normal flora, and then the bad microbes can take hold. For example, antibiotics. Antibiotics will kill your good bacteria. If you take steroids, inhaled steroids, that's going to cause you to get more fungal infection. Infection avoidance. Put yourself in a bubble, right? So if you don't contact anything, then that's actually a bad thing because your body will not recognize anything, any microbes. So that's why we no longer have antimicrobial hand soap because it kills the good stuff and the bad stuff. So that's your normal defense. Name me another defense that you have in your body besides your normal flora. Your skin, yeah. So that's a physical barrier. What else? Your bones, okay. What's inside your bones? What's made inside the bones? Your blood cells. Yeah. So your immune system, that's part of your natural defense as well. So sometimes you have to learn things. Your immune system has to learn things. Other times you just have generic defense or soldier cells. Favorable condition. So why do they tell you to drink cranberry juice when you get UTIs? How does cranberry juice prevent UTIs? So for bacteria to actually cause an infection, they have to take hold. So they have to build a foundation. So they have to make a coating on the inside of your bladder. So cranberry, the sugars in cranberry prevents that. So it just flushes them out. So they don't create that biofilm. Cellular and subcellular. So when they say cellular, they mean pretty much complete life form that is able to self-replicate. That's cellular. If I am subcellular, then I probably don't have everything and I can't replicate and I can't really function unless I'm inside and I use another cell. Right? So for us, pyrons and viruses are subcellular. So these two categories are less than a cell, which means for them to divide and proliferate, they actually have to be using other cells. Then we have bacteria, protozoans, fungus, helminths. Helminths is just a fancy word for saying worms, right? There's different types of worms depending on how many layers, body layers they have. Bacteria. So, do you know what it means to be prokaryote versus eukaryote? So this term only refers to the cells.
Is
So
that like when they carry it and they can give it off?
this is cell structure. So this is microbe cell structure. So for bacteria, they're prokaryote, meaning there's really no structure. Their DNA is just all over the place. But if you're eukaryote, like us, what's in the center of our cells? The nucleus, right? So that's where all of our DNA is housed, right? So they're thinking, hey, prokaryotes, pro for like before. So those were more primitive. They didn't have a really structured cell. As eukaryotes, like euphoria, normal, these are more modern cells. All the DNA is held in the center. So that's the main difference. So bacteria, prokaryotes, makes them very simple, and they can replicate very fast as well. Pretty crazy what they can do. And some of these bacteria are similar to subcellular microbes, where they need a host cell to actually replicate. We'll talk about some of those. Protozoan. Have you guys ever heard of a protist? An amoeba? You guys heard of an amoeba, right? So it has like real no shape. It doesn't really have a shape. It just moves by mobbing around, right? So that's an amoeba. And that can infect your GI tract if you consume dirty water, especially when you travel. Another protozoan is malaria. You guys heard of malaria? All right, mosquitoes carry that. What's a fungus? Mushroom, yeah. There's the mushroom cap. Toadstool, right? Why do they call it a toadstool? Because that's where toads sit on. Duh, no, just kidding. But it can be single cell or multicellular. So single cell are things like yeast, my favorite microbe. You guys know why? Brewer's yeast to make beer, right? So that's what yeast does. They take the sugar from grain and turn it into alcohol. So fungus can also be multicellular, like mushrooms. They're pretty big, or mold. They come in different forms, like a male and female version, or a positive and negative version. And then they kind of couple with their spores, and that's how they multiply. So they have complex life stages as well. And that's yeast infection. Helminths are worms, multicellular. Of course, they're big. Different types: roundworms, flatworms, segmented worms, hookworms. We'll talk more about these parasites. First, viruses. Cellular or subcellular? Subcellular, which means they need to infect other cells, right, in order to replicate. So the two we'll talk about is HIV and hepatitis. So viruses are very, very simple. All they are is a repeating protein structure. So if I get this triangle, right, and all I need to do is a little tiny code for this repeated triangle, now I can make a sphere, right? So I have that protein coat. That's all a virus is, and DNA or RNA material in the middle. That's it. That's how simple they are. All they do is they affect or they infect the host cell. So in the case of HIV, which cells does the HIV virus infect? White blood cells. Which ones? Yeah, T cells. Don't worry, the answer is always on the board, right? So this virus will attach to the T cells, and then it'll inject its genetic material. In this case, it's RNA. So HIV uses RNA. And what that means is it's a special type of virus. It's a retrovirus. So they're kind of weird. So that means it actually has to insert itself into the T cell's DNA. So here we have the DNA of the T cell, and then this virus RNA will insert itself. And now you have a little segment that's virus DNA. So every time this T cell's moving about, it's carrying that viral DNA inside it. So we don't even need virus parts. Right? So that is HIV. What is AIDS then? Full-blown AIDS? Okay, what causes AIDS? Yeah, it's the same thing. So it's when your T cell fall below 200. So that's just the magic number. So if you have T cells that have been destroyed so much that they go below 200, then that is full-blown AIDS, because now you are defenseless, right? You don't have enough soldier cells or helper cells. Hepatitis. What is hepatitis? We have an outbreak here in San Diego. Who's causing hepatitis A? What are they doing? What are they doing? Infection. How? How are they spreading it? Germs with their what? Yes, okay, you can say it. With their poopies. It's all right. Yeah. So we have a homeless situation and not enough public restrooms. So other cities, other countries, there's public restrooms everywhere. But here there aren't many, and there's not porta potties. So what they started doing is going, and that's how you get hepatitis A. It's waterborne. It's usually contaminated food. So if your food service worker doesn't wash their hands, or they have it, they can spread it. Right? So that is waterborne. You usually get, diarrhea, vomiting. Hepatitis B. And all these can affect the liver, hence hepatitis. Hepatitis B, blood and bodily fluids. Hepatitis C, blood and bodily fluids. Hepatitis D, how do you get hepatitis D? First, you have to have hepatitis B. So, if you never get hepatitis B, or if you're vaccinated for hepatitis B, then you'll never get hepatitis D, right? So you need hepatitis B first. So B, C, and D can cause cirrhosis, hardening of the liver, damage, and then lead to cancer, death. What's this yellowing called? Jaundice. Why? Why do you start yellowing?
It's when I know when babies get feces in their mouth.
Oh, that's at birth. All right, so that's similar. It's a similar condition, but here we're talking about the adult case. So your liver is damaged, and now your body's building up toxins. Right? Bilirubin. You guys heard that term for babies? Builds up in their body. So for babies, it's easy. Once their body or metabolism starts going, get a little sunlight, they're done. But as an adult with hepatitis, your liver is damaged, and now your liver cannot get rid of the toxins, and it builds up in your body. Right? And it happens to have a yellow pigment to it. Bacteria, classified by shape and structure. So coccus, round. Bacillus, rod. Spirillum, what do you guys guess? Spiral shape. And then Vibrio, they're like sickle cell shape, just like a C. That's it. If you are staph, then you grow in clusters, right? Like grapes, that's staph. What if you're strep? Strep, yeah, just like strep throat, the bacteria will grow in long chains, right? If there's two of them, then it's just di, right? Or diplococci, right? Diplobacillus. That's just two of them together. So first by structure, and then their growth pattern. So if we swab somebody's throat and it grows in a nice long chain, then we know that is confirmed for strep throat. If you have an infection on your skin and we take a little bit of, we grow it out and we look under the microscope and it looks like clusters of grape, then that is a staph infection, Staph aureus, MRSA, methicillin resistant Staph aureus. Gram-positive, gram-negative. So this has to do with bacterial cell wall structure. So if I am gram-positive, I have a thick cell wall. And the gram stain happens to stain cell wall. So it's positive for the gram stain. So I am gram-positive because I have a thick cell wall. Very good. Now, if I am gram-negative, am I going to stain? No, because I'm negative, right? I'm not going to have any color. So gram-negative, they have a very thin cell wall, but there's this fatty coating, right? That prevents the stain from actually staining the cell wall. So I become gram-negative. What's more dangerous, or what's harder to kill? Gram-negative or gram-positive? 50-50. So use your gram-negative or more dangerous. So now we can scrape your throat, scrape your skin, scrape your infection, look at it, determine the shape and how it grows. We can do a quick gram stain test, and then we'll know which bacteria to treat you with. Right? So that's very important. Acid fast. So some microbes have a coat around them. So we're going to have to dissolve that coating before we can actually see or stain the microbe. So that's what the acid fasting is. Aerobic versus anaerobic. What's the difference? So aerobic is with oxygen, right? Anaerobic is without oxygen. Fluctuative is they can do both, right? So again, my yeast, my lovely, lovely beer yeast, at the beginning, there's oxygen. They use oxygen. Once they use up all the oxygen, there's no more oxygen. So they have to now make alcohol, right? So the beer yeast actually do both. At the beginning, there is oxygen because when you mix everything up, there's no way you can get all the oxygen out. Special group of bacteria. So these are those kind of like subcellular microbes. So these are like subcellular bacteria. So a lot of these will need a host cell to replicate and to multiply and infect other cells. So some of these you guys probably heard of. Rocky Mountain fever, right? Have you guys heard of Lyme disease? Where do you get Lyme disease from? Ooh, what causes Lyme disease? So we know it's from a bacteria, insect, yeah. Which insect? Ticks. All right. So if you go into the bush area, sometimes ticks are on deer, and then you get the tick on you. It'll inject not just one bacteria, but it's like hundreds and hundreds of different bacteria that causes Lyme disease. So don't go out hiking unless you spray and cover up and know your risk factors. So here in California, it's lovely warm weather, so we have ticks almost all year round. When it's colder, then they don't come out. It's safer to hike then. Drug-resistant microbes. What causes drug resistance? Antibiotic. What? I thought antibiotics killed them. So misuse and overuse. Give me an example of overuse. That's misuse. So until gone, right? Usually you're supposed to use it until all the bacteria is dead, but once the bacteria goes down a little bit, people feel a little bit better, and what do they do with the meds? They stop. Which ones are the ones that survived? After taking antibiotics, It's the strong ones that survive, right? So which ones are the ones rebounding? The strong ones. So if you don't kill them all the first time, it might come back even more. Also, you have to know your antibiotic history because, like I said, you're an ecosystem. So if you use the same antibiotics over and over and over, what's going to happen in your own ecosystem? Yeah, the microbes in your own body gets used to it. That is the big problem with nosocomial infection. What is a nosocomial infection? One that you pick up in the, yep, when you're admitted, right? So that means they have that problem because which antibiotics are they using in their facility? Are they using the same ones over and over and over? Are they developing superbugs? So you got to know your microbe in your building, in your facility. Rotate your antibiotic use, rotate your microbial agents as well. Overuse. Give me an example of overuse. We got misuse pretty much down. Where's most of our antibiotics used? At home? No, not even for human. We have a meat addiction in this country, right? So if we have cows, pigs, chickens, monkeys that are sick, are they growing bigger? The last one was a joke, no one got it. So that means if your chicken's not fighting an infection, then it's growing, right? So might as well feed it some antibiotics so it doesn't get infections. But they do the same thing with arsenic. You guys know that, right? They used to feed chicken poison. That's because it'll kill off some of the microbes and infections they might spread on. But it's just a mild case of poison. I don't think it gets into the meat for human, right? Right? Maybe. I don't know. So chickens got their rights. They got extra room. I don't know if they got their chemical rights, right? So risk factor for infection. Definitely age, immune system. If you have HIV that attacks your immune system, then yeah, you're gonna be susceptible to even simple everyday infection. What did I say about these for hand soap? They are now outlawed, right? Because this was the problem with polio. When kids got polio, usually they didn't have a problem. They were able to fight it off. It got worse if you caught polio when you were older. So during the 50s, that was when everyone was cleaning everything. We had that chemical revolution. So we weren't exposed to these microbes at an early age where we can fight them off. So we got paralysis and then polio, the March of Dimes, everything that came from it. So we're learning no more antimicrobial hand soap to prevent superbugs. So some things your facility might have to do is rotate antibiotics and rotate their cleaning agents. So they might have to change the pH, maybe clean with acid soap 1 month and then next month clean with basic soap, right? Just to stay one step ahead and prevent superbugs. Protozoans, eukaryotes, human cells. Are we prokaryotes or eukaryotes? Do we have a nucleus? Yeah, so very similar to human cells. And a lot of times that makes it kind of hard to fight because if it's bacteria or fungus, we can kind of detect them easier. But eukaryotes, they kind of sneak past our defenses sometimes. So an example is the amoeba that causes pretty much GI distress, diarrhea, vomiting. And in developing countries, all you really need is an IV to rehydrate you, but they don't even have access to that, and most of them will die. Catch something for breakfast, and then by dinner time, you die. So that's the tragic case with a lot of people. They don't have safe drinking water. So is that a right or a privilege? Is water a right or a privilege? It's a privilege? Really? I have to earn water? Sorry, you're not going off to do any humanitarian work, are you? So there's a lot of people who go to third world countries and they help them build wells and safe drinking water. And that just prevents tons and tons of premature death. Fungi. So fungus is one, fungi is many. So just like bacteria, right? Bacteria kind of lives forever because they just make a copy of themselves. Yeast kind of does the same thing, they just bud. So they just make a little friend and then their friend buds off and then it's a clone of themselves. So technically, yeast lives on forever as well because they just make a copy of a copy of a copy. So that's single cell structure. Here for things like mold and mushroom, they're multicellular. So they'll make spores that'll have different sex, like I'll be a plus sex, I'll be a negative sex. It's not really male and female, but they can grow as that spore, and then when those spore things grow up, they'll combine, have sex, and then they have different and very complex life cycles. Right? You guys heard of a truffle? No, the most expensive food in the world. Truffle oil? Truffle chocolate. Okay. Truffle fries? No. Okay. So so that is a fungus that grows underground. And usually if it grows above ground, it makes a mushroom, right? So it depends on the condition. If the conditions are right, I'll grow into a mushroom. If the conditions aren't right, then I'll grow underground into a truffle. Multicellular. So now we're getting into worms and insects, mites. So parasitic worms, many types. If your kid complains about an itchy butt at night, it might be parasites. Because kids are clean, right? They taste the sand to make sure it's clean before they swallow it. They taste, everything in their surrounding, make sure it's clean before they swallow it. So if you have pets and the kid gets into the litter box, there you go. There's one way you get the larva of these parasites into your digestive system. So what's going on is these worms, they don't want to lay the eggs inside because they don't want to fight with their kids for food and nutrients. So they poke out the anus, lay their eggs, and your kid has an itchy, itchy butt. They scratchy, scratchy, scratchy. And then while they're sleeping, and then that life cycle continues. They suck their thumb, they get the larva back in their digestive system. Rinse, repeat, right? So itchy butt, make sure you come in with a flashlight, do some detective work. Well, now you're not a parent yet. Don't worry. When you call the doctor, my kids complain about itchy butt every night. Then the doctor's going to tell you to check every night, and it's the same thing. Kids are, like I said, very very clean. They make sure they test. It could be anybody, because it's cross contamination. It doesn't have to be direct, right? Insects, ticks we mentioned, give you Lyme disease. And that's a host of bacteria that it's being injected. Lice feeds off of your blood. So anytime they're looking for lice in the school, that's because, is it random testing? Somebody reported a lice outbreak. So if you had a lot of lice checks in your school, grade school, that means there was a lot of outbreaks. So we had at least one lice check a year. Sometimes there was more, so I think anytime there was an outbreak. Scabies. What are scabies? Little bumps. Are they pimples? No, they're little insects that burrow into your skin. So it makes a little mound that looks like little pimples, but it's actually insects. And don't worry, your whole family will be treated. Burn everything. You do a shampoo. A body shampoo. You cover it and you use that overnight, and then take a shower the next day. Hopefully that kills off all the scabies. Right? Treat the whole house. Malaria. What's passed on from malaria bites? You guys heard of this guy?
Yeah.
So it was only the last few years we had pockets of West Nile, and now it's everywhere in the United States. All right. What's this guy? What did it cause? Microencephaly, right? The small head. Right. So that's mosquitoes. And then of course, malaria. Right. You guys heard of malaria? So malaria has multiple life cycles as well. So it affects your blood and then your lungs, and then you'll have fevers and chills, So mosquitoes are bad. How do you prevent mosquitoes? No water, right? So that's how they breed. They lay their eggs in the water. So if you have standing water, then just turn it over, cover it, and then no mosquitoes. But guess where we are situated? Right here next to the marsh. So tons and tons and tons of mosquitoes. So don't go out in the early morning and the late evening, because that's where they're most active. Right? But that's when it's the best weather to do your jogs and runs and walks, right? So just spray. Unfortunately, the chemicals are the ones that work the best. But would you like to have Zika, West Nile, malaria? Now, you guys heard of sickle cell malaria? Sickle cell. So sickle cell malaria, if you're a carrier for the sickle cell trait, you're immune to malaria. So in areas where there's a lot of malaria, there's also a lot of sickle cell trait because you survive. Because if you're a carrier, you live. It's unfortunate that if you gave your kid sickle cell, then they might not live. But you live, so it's okay. How infections are diagnosed. So examine the patient. We'll get a specimen. So if it's skin, we'll swab it. Throat, we'll swab it. Sore, wound, whatever. We'll get a sample of it. We might have to grow it out. That's what we're doing with our blood plates. Why are we using blood plates? And these are 5% blood plates. So we're using it for qualitative analysis. So if there are pockets that are clear, then we know that the microbe's able to eat the blood. They're hemolytic. So that's why we use 5%, and in this case, it's sheep blood. Can we grow viruses? Pretty hard. A mold will grow on these, and fungus will grow. Some protozoans are really hard to grow. Culture and sensitivity. Sensitivity to what? To who? To air, okay. Yeah, sensitive to antibiotics. So what we'll do is we'll grow out a plate, we'll divide the plate, and then in different areas, we'll apply different antibiotics so we know which one will kill it. All right, so that's what we mean by sensitivity. We grow out the bacteria, and then we treat it with different, hopefully killing agents for them, antibiotics, to see which one works. So we have to ID. That's the most important thing when treating an infection. We'll give them a broad spectrum antibiotic, an antibiotic that will work, maybe not the best, but it'll cover a lot of microbes. Hopefully it brings down the infection. And then once we ID and know exactly what it is, then we'll give you the specific antibiotic, a narrow spectrum antibiotic.
So is that why they use penicillin a lot?
Penicillin works for most of our infections. So penicillin, PCN, is good for gram positive. So very effective. So a lot of our common everyday infection, gram positive. A benign infection, and then a weaker antibiotic. So once you get into the gram negative, they're going to treat you with different types, stronger antibiotics. Collection. So you guys will be doing some collections. So you guys can do swab, you can do contact collection. So we'll divide out 2 for each of you guys, and just label where you guys want to go, and then just sample it out. Once you're done, just tape it. Which way do we store the agar plates? Right side up or upside down? Suspended, right? So make sure the agar's up, because if there's any condensation that drips, it drips down on the cover. So if you have it right side up, any condensation on the cap or lid will then just drip right on your sample. Right? So that's why we incubate them upside down. We don't have anybody with an infection, so no suspected, so we won't do any throats. You guys done urine analysis, right? Where you guys spun it down and you guys viewed it. So we don't have to do that in this class. So you guys viewed all the crystals, all the different cells. Sputum, what's the difference between sputum and saliva? Spit. Saliva's that watery stuff, right? We don't want to collect any of that stuff. We want that thick mucousy stuff. That's where all the microbes might be. Because saliva, it just comes out of you. There's no sample. There's nothing in it. You'll dilute it. And then if you need to swab the wound, transporting, most of the time we'll look at it in-house. So just store them or fix them as prescribed by your physician or your policies and procedures. If need to, just package your specimens appropriately for pickup. So that means we're going to need some preservatives in these samples, because as soon as you take it out of the body, it's either going to get infected, start growing really, really fast, or it starts to break down and you kill your sample. Right? So maybe you need to preserve it, maybe you need to freeze it, or to stop it from growing. What is NS? Normal saline. Normal saline. And what does that mean? It is 0.9% sodium chloride. So for our wet mounts, remember that's what we use to bathe the cheek cells and the cheese sample, because NS is mostly balanced with life and it won't damage the cells. Apply your cover slip. If you need things to be dissolved like keratin, so if this is tissue from the body and you have skin or nail, like nail fungus, and you need to remove the keratin, the protein that is skin and nails, then you'll bathe it, 30 minutes in a basic solution. So it's not acid, it's basic. And that dissolves out the keratin, and then you can just have the cells left over, and it's easier to view. We'll do gram stain week 3. So this week we'll do our culture plates. Hopefully we'll have growth by should have growth by next week. If not, I'll do some plates during the week, and then we'll sample those plates on week 3. We'll do gram stain at that time as well. CFU stands for colony forming unit. Theoretically, one bacteria will divide into 2, which then will divide into 4, and exponential growth. So every dot is one bacteria, right? And the one next to it might be the same, or it might be a different color. So that's what they mean by a colony. One colony forming unit, and usually comes from one bacteria. So on the agar plate, if it has something that is dark red versus a bubble that is clear, it can tell me which one can use blood, which one can eat blood. That clear one, right? Because it dissolved the blood. It was able to be hemolytic and use it. So these guys here, they might be using that blood. so we can determine qualitatively the type of bacteria it is. All right, not too bad. So if you weren't here week 1, you got to catch up with your wet mounts. So do cheek, do some cheese, do blood, cover slip, NS if you need it. And then this week, everyone grab 2 plates. Get the Sharpie, put your name, date, and your sample. Get your sample on, either with the swab or direct contact. Come back, tape them up, and we'll incubate them. And then do your dry mounts and right stain for blood feathered.